Hello, everyone. So welcome to elementary statistics. Today, we're going to start with chapter 1. So we're kind of just going to introduce some basic concepts. So obviously, this is a statistics class, so we should probably start off with the definition of what statistics is. So statistics is the science of conducting studies to collect, organize, summarize, analyze, and draw conclusions from what we call data. So why do we need to study statistics? So there's various reasons. So one of the first ones being you need to be able to read and understand the various statistical studies performed in your field. So say, for instance, you're in the medical field, you need to know how to read a medical report. So maybe what someone's blood pressure readings are, how do those compare to normal? You may need to conduct research in your field. So that could be like you're in animal science, you're in plant science, you need to conduct some research using some statistical analysis methods, or even some software. And the last one, you can also use statistics to become better consumers and citizens. So now that we've given a definition of statistics, we want to introduce two branches of statistics we will focus on in the course, so descriptive and inferential. So the first introduction definition we'll talk about is what a variable is. So a variable is a characteristic or attribute that can assume different values. And what these data values we're talking about are the values that random variable can actually assume. So the values it can take on. And variables whose values are determined by chance are called random variables because chance is random. So to infer validly that the results of a study are applicable to a larger group than just the participants in the study, we must also define the population and the sample of participants before we do any study. So the population versus the sample, what is the difference? So the population is the collection of all individuals in the population of interest, and a sample is a smaller collection from the population of interest. So basically, a sample is a collection of individuals from that population. So how I refer to it, a sample is a subset of the population. So now we want to give the definitions for descriptive and inferential statistics. So these are the two main branches we're going to talk about this semester. So the first half of the semester, we'll talk about descriptive statistics, and the second half, we'll talk about inferential. So descriptive statistics consist of the collection, organization, summarization, and presentation of data. So this is by computing various probabilities, by doing various statistical computations on the calculator, even by hand. So, and then the second one, inferential statistics consist of generalizing from samples to populations, performing estimations and hypothesis testing, determine if relationships exist among variables, and then using those relationships to make predictions. So this inferential part we will look at in chapters 7, 8, 9, and 10. So try it out. So now we want to determine whether descriptive or inferential statistics were used. So number one, the average price of a t-shirt at the student union is $32. So we're basically just describing how much that shirt cost on average at the union. So we're just presenting the average price. So we will call that descriptive. So let's look at the next one. So based on the current inflation rate, we project the median price for an apartment in Stillwater next year will be $450. So we are projecting the median price for the upcoming year based on what we have right now as far as inflation goes. So that would be considered inferential. So the next one, the average price for a dinner at an excellent restaurant is $45. So basically just summarizing the price of a good dinner. So that's just descriptive statistics there as well. So now that's all for section 1.1. Now I want to move on to 1.2, where we talk about variables and types of data. So the first thing we want to do is match each word to its correct definition. So the first one we'll look at is qualitative variables. So qualitative, we think of categories, right? So qualitative variables are variables that have distinct categories according to some characteristic or attribute. So then we'll move on to quantitative variables. So quantitative quantities. So it involves numbers. So variables that can be counted or measured. So counted, quantitative. Continuous variables. So continuous, it can take on an infinite number of values. Think of it like the interval 0 to 1. It's continuous and it can assume an infinite number of values between any two values. So that's what continuous is. And discrete, these assume values that can be counted. So we can count the number of values it can take on. Then we look at this little classification chart on the next page. So it says the classification of variables can be summarized as follows. So we have variables up here. We can break them into qualitative and quantitative. And within quantitative, we can even break those down into discrete and continuous. So let's look at some examples for each of these. So first we'll do for qualitative variables, so just qualities. So you could talk about the color of shoes in your closet, the color of your car, your cat's breed, the color of your cat. Some quantitative variables you can talk about, so anything that can be measured or counted. So the number of pets you own, the current bank account balance you have, even the current body temperature. So within those quantitative variables, we can have discrete and continuous. So discrete, we can count them. So the number of workers in a company, you can count the number of workers in there. Shoe sizes, there's only a certain amount of shoe sizes. You can count those like 1, 2, 3, 4, 5, 6, 7, 8, and 9. The number of touchdowns in a game. Then we look at continuous. So these ones are kind of tricky because continuous is a little bit different. So time, we always consider time to be continuous because it's never ending. So time is a continuous variable. Height is also a continuous variable because you can take on any height in between, say 0 inches to however tall the tallest person can be. And you can use the same idea for saying that weight is a continuous variable as well. So now that we've introduced some types of variables, what we want to talk about are boundaries. So we want to look at some variables, the recorded values, and the boundaries we want to create around them. So since continuous variables must be measured, values are usually rounded to the nearest given unit. For example, if the temperature is 86 degrees, this could mean the actual temperature is between 85.5 degrees and 86.5 degrees. So these two values, 85.5 and 86.5, are what we call the boundaries. So now we want to kind of look at some examples of boundaries and how we're going to find them. So the first one, length. So the recorded value is 15 centimeters. What we want to do to create these boundaries is, so this right here is to the nearest whole number. So we want to go to the first decimal place, the tenths, and take half of that. So we're just going to take half off of that value. So if we take half off of 15, that would give us 14.5. And then if we add 0.5 to 15, that gives us the upper boundary of 15.5. So then using the same idea here for time. So time is to 2 decimal places. So we're going to go to the third decimal place and take a half off of that decimal place. So if we do that, we would be subtracting 0.005 to get the lower limit. So that would be 0.425. And then similarly, we would add 0.005 to find the upper limit of 0.435. So since we had 2 decimal places here, we're going to do 1 here. So mass is measured to the nearest tenth of a gram. So what we want to do is go to the second place and take off a half. So we'd be taking off 0.05 and adding 0.05 to find the boundary. So when I take off 0.05, that gives me 1.55 grams for the lower boundary. And when I add 0.05, I get 1.65 for the upper boundary. So we'll see this again in chapter 2 when we talk about different types of graphs. So we'll talk about class boundaries whenever we get to histograms, and we'll also talk about some class midpoints and stuff like that. So how are variables categorized, counted, or measured? So we're going to talk about 4 different levels of measurement. So the first one being the nominal level. So the nominal level of measurement classifies data into mutually exclusive categories in which no order or ranking can be opposed. So it's basically just categorical variables that have no meaningful order. So we could talk about college majors, so what your major is, blood type A, B, O, eye color, blue, brown, green. So in all of these categories, there's no ranking or no order assigned to them, and they're basically just categories. So then we're going to call these types of variables nominal. So then the next one we'll talk about is ordinal. So ordinal is the level of measurement that classifies data into categories that can be ranked. However, precise differences between the ranks do not exist or are meaningless. And a little note here, the difference between two levels cannot be assumed to be the same as any other two levels. So ordinal. So these are basically categories, but they have a specific order or ranking to them. So we could talk about economic status. So whether you're high, middle, or low class, you could talk about college classification. So if you're a freshman, sophomore, junior, or senior, another example would be student satisfaction on surveys. So if you're not satisfied, slightly satisfied, highly satisfied, so stuff like that. So then the next thing we'll talk about is the interval level of measurement. So this ranks data and precise differences between units of measure do exist. However, there is no meaningful zero. So this zero is basically kind of like a baseline. So note the distance between each interval is the same along the scale. So and a quick examples we'll talk about are Celsius. So that's just a measure of temperature, pH, and another one is credit score. So the last level we'll talk about is the ratio level of measurement. So it basically has all the characteristics of the interval measurements, but a true zero does exist. So the way I like to think about it is if you can compare two different members of the population based on this measurement that you're taking, then it will be a ratio level of measurement. So we could talk about flow rates. So the rate of some fluid going through a certain system, the concentration of some, say, chemical that you're working with in a lab. You could also talk about the dosage amount of a drug. So there's different dosages, 5 milligrams, 10 milligrams, 15, and so on. And then the 0 milligrams there just represents you're not taking the drug at all. So there is a meaningful zero there. So then the next thing we want to talk about is data collection and sampling techniques. So recall that data can be collected from a subgroup of the population, and we call this a sample. How can we collect sample data? There are 4 methods we will discuss in this chapter. So a random sample is the first one. So a random sample is a sample in which all members of the population have an equal chance of being selected. So this equal chance is what makes it random. So say you put all of these names in a hat and you draw 1 or 2, so that would be considered a random sample because everyone in that hat has an equal probability of being selected. So then the next one we'll talk about is what is known as a systematic sample. So a systematic sample is a sample obtained by selecting every kth member of the population, where K is a counting number. So that just means 1, 2, 3, 4, 5. So say, for instance, in the class that we have right now, I took every 5th member of the class, and then I wanted to see what their grade was in the class. So that would be a systematic sample. And every kth member would be every 5th student in the class. So the next type of sample method we'll talk about is stratified. So stratified is a sample obtained by dividing the population into stratum or groups according to some characteristic relevant to the study. And then from each of those groups, we take random samples. So some stratum we could talk about is one that we mentioned on the previous page, classification for the university. So we could take a group of freshmen, and then from that freshman group, we could take a random sample and then measure something we want. So then the next one is a cluster sample. So a cluster sample is obtained by dividing the population into sections or clusters, and then selecting one or more clusters as the member of the sample. So notice there are some similarities between stratified and cluster. We're both dividing the populations, into certain groups. However, the groups could be more or less homogeneous in a cluster sample compared to a stratified. So that more or less homogeneous means they could possess a certain characteristic similar to all the others in the sample. So now, since we're taking just a subset of the population, we can always expect to have error. And this error that we expect from the sample compared to the population is referred to as sampling error. So sampling error is the difference between the results obtained from a sample and the results obtained from the population from which the sample was selected. So then we'll talk about non-sampling error. So this is when the data are obtained erroneously or sample is biased. So non-representative is a word that we'll talk about. So non-representative means it just doesn't represent the population as a whole. So now that we just mentioned non-representation, we're going to talk about non-representation bias. So non-representation bias occurs when you take a sample that is not an accurate representation of the population you are interested in. So it doesn't represent the population. So non-response bias occurs when you attempt to take a sample, usually a convenience or volunteer sample. So convenience, this is just use sample who is convenient, and volunteer is they just volunteer to take place. So, and we have a significant percentage of individuals who do not respond. So they don't respond, so hence non-response bias. So this last section we'll talk about is section 1.4, and it's about experimental design. So we're going to talk about observational versus experimental studies and what is the difference between them. So in observational studies, the researcher merely observes what is happening or what has happened in the past and tries to draw conclusions based on these observations. So observational study, we're basically just observing. So the next thing we'll talk about is experimental studies. So in an experiment, at least one predictor variable is controlled or assigned by a researcher team, and the independent variable is the one that is being manipulated or changed. And we're looking to see how this affects the dependent variable or the response variable. So there may or may also be uncontrolled observed variables. So first thing we'll talk about is the definition of a classical experiment. So a classic experiment assigns variable values on an individual by individual basis. So it just means this person gets this treatment, this person gets this treatment, this person gets this treatment. So, however, in a quasi experiment, variable values are assigned on a group by group basis. So we could look at classrooms or dorm floors and say this classroom gets a certain treatment here, this dorm floor gets a certain food type here, this classroom gets a certain snack this day or something like that. So the administrator is actually who knows the treatment assignment, and subjects may or may not be blinded. So that just means whether they know what treatment they're getting or not. And also administrators of treatments may or may not be blinded. So if the administrator doesn't know the treatment that the subject is getting and the subject doesn't know the treatment that they're getting, we refer to that as a double-blinded experiment. So now the next thing, so on the other hand, a confounding variable is one that influences the dependent variable, but was not separated from the independent variable. So it's just like something that influences the outcome, but wasn't directly related to the study. So, and then one other thing we'll mention is a survey is a type of observational study that yields self-reported variables at a single point in time. So just like you take a survey at the end of this class to see what your satisfaction level is. So that is totally voluntary and it is at a single point in time. You're telling me, how did I do over the semester? So then we'll talk about the groups in an experiment. So a treatment group receives a new idea. A control group receives the current standard treatment, which may or may not be nothing. So say this treatment group gets a new drug and this control group gets the one that they have already been receiving, or they could get what is known as a placebo. So now that we've introduced the word placebo, we want to basically mention what that is. So a placebo is a neutral treatment that appears the same as the actual treatment, and control groups often receive a placebo because people psychologically respond to the perception of a treatment. So they basically think something is happening or they're improving, but in reality, they're not. So this is what we refer to as the placebo effect. So now let's look at an example and see if you can tell the difference between observational and experimental studies. So the first one, a researcher stood at a busy intersection to see if the color of the automobile that a person drives is related to running red lights. So we're assigning treatments here to see how the vehicles were affected. No, but not. So they're merely just watching to see what car runs the most red lights. So that would be observational. So the next one. Subjects were randomly assigned to two groups, and one group was given an herb and the other given a placebo. After 6 months, the number of respiratory tract infections each group had was compared. So in this case, we had two groups. They were assigned a treatment. So one was given a new herb and one a placebo. And then we are measuring something over a period of time. So this is an experiment. So since this is an experiment, then we want to identify the independent and dependent variables for the study. So the independent variable is the one that is being manipulated or changed. So in this case, what is changing on an individual by individual basis? It's whether they are getting the herb or the placebo. So we would say the drug type is the independent variable, or you could say type of treatment. And what are we measuring? We're measuring the number of tract infections each group had at the 6-month period. So that would be our dependent variable. So now, what are some possible confounding variables for the study? So something that could influence the number of respiratory tract infections besides these treatments we got. So you could talk about family history, whether you've had a respiratory tract infection before. So those are just two possible ones. You could also come up with many more. So now that we finished up the section, we want to talk about a little bit of a review over chapter 1. So determine whether descriptive or inferential statistics were used. So last semester, the average score for exam 1 was a 70. So he's just describing the average score from last semester. So that would be descriptive statistics. The second one, based on the scores from previous semester, the average grade on exam 1 this semester will be an 80. So here we're using educated guess from the past to infer something about what's going to happen in the future. So this would be considered inferential. So the next thing, determine whether qualitative, continuous, or discrete variables are in place here. So the number of students in attendance. So we can count the number of students in attendance. So this would be a discrete variable. Your college major. So that's based on some characteristic or attribute. So it's just a quality. So it's going to be qualitative. So the time it takes to drive to Oklahoma City from Stillwater. So this is talking about time. And like we said earlier, time is never ending. So it's continuous. So classify each as nominal, ordinal, interval, or ratio. So hair color, that's basically just a categorical variable that has no meaningful difference between levels. So that would be nominal. So the temperature in every OSU building at 8 a.m. So that could actually be interval or ratio, depending on if you want to compare the temperatures in the buildings, but usually we would use interval there. The age of all mathematics professors at OSU. Again, this one could be interval or ratio, depending on if you're just talking about a single individual or if you want to compare the age between two professors. And then the last one, teacher evaluation rating. So these are ordinal. We talked about that earlier. There are categories involved. So satisfied, highly satisfied, not satisfied, and there is some order there. So, and then the last part we'll talk about is this example where we're talking about an observational or experimental study. So researchers randomly assigned 10 people to each of 3 separate groups. Group 1 was instructed to run a treadmill for 10 minutes. Group 2 was instructed to run an elliptical for 10 minutes, and group 3 was asked to do 25 minutes of lifting weights. Then after each of the groups finished their workout, their heart rate was measured and recorded. So was this an observational and experimental study, and why? So it's asking, did we assign different people in this study treatment levels to see how it affects some outcome? And yes, we did. We assigned them to group 1, 2, and 3. And then based on it, that group, then we wanted to measure their heart rate at the end. So then this was a experiment because treatments were assigned, and the treatment was based on whether you're in group 1, 2, or 3. So what is the independent variable? So what is being changed or manipulated to see how it affects the outcome variable? So in that case, it would be what group you were in. So what exercise you were doing. So and what is the dependent variable? So what are we measuring? We are measuring the heart rate at the end of the workout. So what are some possible confounding variables in the study? So something that could influence your heart rate besides the actual workout you did. So you could talk about how often you go to the gym, whether you have heart problems, your health, your age. So those are just a couple ones. There also could be a couple more that you could think of. So that is actually all for chapter 1. So I will see you all in chapter 2.